Hello, this is Core Practical 1, looking at cells for biology. So what equipment we've got is a light microscope here. We have some stain, which we're going to use iodine. We have a knife for cutting the onion, some tweezers for pulling back a bit of the onion skin. We've got a slide, and we have got a cover slip. So the first thing that we're going to do is get a little bit of our onion to look at the microscope. So I'm going to cut the onion, and I'm going to use the tweezers to peel back the skin to get a nice transparent or clear or see-through section of skin from the onion. Right, I've got a good chunk of that now. If I put that onto a microscope slide and then put that under the microscope, because it's transparent, you wouldn't be able to see the cells. You would be able to see the outline, but you wouldn't be able to see a very good, clear image. So what we're going to do is put our sample or specimen onto the microscope slide. We're going to get a bit of the stain, which is the iodine in this case, and that's going to make the cells more visible so we can see them, just one drop. And then we're going to get the cover slip and put that over the side and drop it down. I'm going to place the slide on the stage of the microscope. Hold it in place with the clips. There are two different lenses. There's the eyepiece lens, and these are called the objective lenses. The eyepiece lens magnifies things by times 10, so that makes things 10 times bigger. The objective lens make things, this one is 40 times bigger, 4 times and 10 times. It will tell you actually on the objective lens. So if we wanted to work out how much bigger it's made it, it's not adding them together, we need to multiply it together. So I'm going to start on the lowest magnification, so this is 4. The objective lens is times 4. The eyepiece lens is times 10. So this is going to make the image, when I look down into it, 40 times bigger. I'm going to use the focusing wheel here to move the stage, and that is going to bring the image into focus for me. I always bring it up as far as it will go without touching the objective lens. Look through the eyepiece lens, and then you're going to slowly bring it down until it comes into focus. There you go. If you needed to, you sometimes have a fine focusing wheel, and if you turn these, it would bring it into a better focus for you. After you have got a times 4 image, or times 40, we'll move on to the next objective lens, which is times 10. I'm going to do the same again. Now we're going to move up to the next objective lens and do the same again. And that's it, it's easy. The only other thing that you might need to know is if you zoom in on this question, you might be given the magnification of a picture and then asked to work out what the actual length is. So if the picture is 8,000 times bigger, the real length must be 8,000 times smaller. So you need to divide by 8,000. And then what they like to do is trick you. So they've given it in millimeters, and they want you to state the answer in micrometers, which is 1,000 times smaller than millimeters. So then you would divide your answer again by 1,000. That's it. Well done, everyone. Thanks for listening.